thank you very much for this opportunity to talk about Xcell and the avatar and the exciting applications that we have going on with our customers. So thank you all for joining us, and I'd like to talk a little bit about how we approach cell biology and cell culture. As many of you know, there was a lot of work done by Greg Semenza discussing hypoxia. And as we all know that in the atmosphere around us right now, we are at 21% oxygen. However, as soon as you get into the body, that level of oxygen drops off and essentially gets into a hypoxic state compared to the atmosphere. And as you see, even when you get down to things like arterial blood, down to venous blood, all the way down to muscle, brain, we're at a fairly hypoxic state, and solid tumors being very hypoxic, around 1%. When our founder, James Lim, was working in the lab and trying to grow primary tumor cells, one of the things he found was that actually just considering hypoxia was not enough to grow cells. And what he found was the work of Rakesh Jain at Mass General in Harvard, that when you look at the cells in their microenvironments, especially the primary cells that are coming out of the body, you see that they actually are under a pressurized environment. So the normal breast here is 0.02 psi, and when you get to a breast carcinoma, you see that that pressure jumps more than 10 times. So the idea is that the cells themselves are seeing not only hypoxia, but a pressure. Sorry. So you see here, when we go from the atmosphere to the lung, you again see the drop off of the oxygen level, and you actually see a slight increase of the pressure being the structure of the alveolar sacs. And then as you get further out into the body, you see again, mimicking what Rakesh Jain and Greg Semenza show, that the oxygen tends to drop off. And again, some of these structures like the brain, the bone, and then ultimately the tumor have a much higher pressure. And again, when we're looking at these cells, trying to culture them in vitro, the idea of the avatar is to get them in a microenvironment as close to the microenvironment from which they came. These are the applications that we have already actually worked on. We've divided it into four different categories, but immune cells, stem cells, oncology, tumor, and then others. So we've actually had some success in customers coming to us with very specialized requests and actually been able to do it with our pressurized pressure and hypoxia in the avatar. So one of the first customer data that I can show you, it's blinded and we are under NDA, so I cannot tell you what company this is, but I can tell you about the data that they did, the data they produced. So their problem was they were working on a drug for erythrocytes, and they needed their erythrocytes in a specific cell profile or state and identify biomarkers of the target cell population. And this was after 18 days of culture. But what it required was a very low inter-user variability. This was to generate a predictive and reproducible result that they could present to regulatory agencies. So the initial steps, they use multiple avatar conditions to evaluate where the biomarkers were being expressed, and they assess the biomarkers by panning using a protein chip. At the end of this process, basically, they identified the biomarkers and they did an initial run for robustness of assay and saw 6 parallel culture replicates performed by 2 different operators and ended up with 21 total replicates over 1 month. And as you see here, the overall coefficient of variance is under 5%, which is fantastic for cell culture. So this was their initial steps. They then went on to long culture stability with the avatar, and then transition the cell state to be controlled, looking at the microenvironment, and then using multiple samples, multiple users to see if they could handle this SOP over multiple sites. And again, what they found was they had a very tight CV, again, under 5%. So the end point of this, and again, we are under NDA, but the avatars are now integrated into an improved SOP for QC release testing of this particular drug. And we actually we're a little bit past this step, but the avatars have now additional avatars have been purchased and integrated into their global CRO sites, again, to implement this standard operating procedure, which they developed on the avatar. So very exciting for us. We didn't realize we were going to get into a regulatory part of the marketplace this quickly, but this was a need from this company and went forward. So moving on from the blood cells to the immune cells, we see here, we had another company coming to us that was building a CAR T platform, but the company was using this in a standard incubator. And they approached Xcell Bio to answer the question, would the CAR T work in a tumor and microenvironment? So the CAR Ts were blinded to us, and they were provided to us by the customer, as were the liver tumor cells, and they were processed using a protocol that we had for disassociation of solid tumors and then plated on glass bottom plates. All of these were actually grown in a standard incubator until the final step. And the final step is utilizing in the avatar, as opposed to the standard incubator, 5% O2 and 2 PSI. This was the conditions that we found that were found to actually grow the cancer cell the best. So this was taken as the tumor microenvironment that these CAR Ts would actually encounter if they were put into the body. So what we found was, oops, hopefully, sorry. There we go. So what you see on the right, on the left is the standard incubator. So this was the final co-culture. And as you see here, the CAR T in the standard incubator works quite well and starts to attack and kill the cancer cell. Now, if you note that this is a 30-minute time-lapse movie, as opposed to this side, which is an 8-hour time-lapse movie. This is, again, the tumor microenvironment where we have 5% oxygen and 2 PSI, which was the conditions that the liver tumors grew the best under. And as you can see, several of these cells just go through. The cells are not killed, and there's appreciably no cell death even after 8 hours. So the conclusion was that the CAR T was much less effective in the tumor-like microenvironment at 5% O2 and 2 psi. Now, although this was negative data, the companies found this to be incredibly important because they want to be able to stop a product before it becomes too far down the drug pipeline. And if it's not going to work, it's better to know this early. And one of the things that Avatar was able to give them was a predictive model for how the CAR Ts would work in an in vivo setting. To kind of further go on with this research, we were working with Larry Fong's lab at UCSF, and they wanted to take a look at the primary T cells in a tumor microenvironment. So as you see here, the problem that they were asking, the question they were asking was, do the T cells from PBMCs behave differently when grown in a standard incubator versus an Avatar? And so again, they were mimicking the optimal settings for their cancer cells, which were 1% O2 and 2 psi. So they are using normoxia, and let me get the laser pointer. So they were using normoxia at 20% O2 and 0 pressure, and the tumor microenvironment, which is 1% O2 and 2 psi. And I should say, I neglected to tell you that the 2 psi is 2 pounds per square inch above atmospheric pressure. So whenever we refer to a psi number, it is above atmospheric. So this one is at atmospheric. This is one. This particular microenvironment is at 2 psi above atmospheric. The culture was run for 3 days, and then they measured these readouts. The first question they asked is, when you take these cells and put them into pressure and hypoxia, is there a large die-off? And this data here answers that question quite nicely. Basically, the answer is no. There's no large die-off, and the cells are actually living and changing from the data I'm about to show you. So no skewing of the data because of a large die-off of the cells. As you see from here, Chiara looked at the cytokine release of activated lymphocytes in culture. And the way to read this table is, this is the tumor microenvironment versus the standard incubator. And so in the tumor microenvironment, interferon gamma, TNF alpha, and IL-10 are all very strongly downregulated in the tumor microenvironment. So there was a generalized decrease in the function of these cells. Going forward, looking at metabolic markers, CD71 and 98. So CD71 is a transferrin receptor, and CD98 is an amino acid transporter. All of these were actually lowered in expression. Now, GLUT1 was a positive control for them because GLUT1 is a hypoxia indicator. So the 1% oxygen and having GLUT1 increase is actually a positive control that the hypoxia was definitely present. Going on from there, looking at CD4 positive cells, and again, this is tumor microenvironment versus standard incubator. And this is day 1 is compared to day 0, day 2 is compared to day 1, and day 3 is compared to day 2. As you see with the tumor microenvironment, PD1 is significantly increased over the 3 days, and ICOS is significantly increased. However, TIGIT and TIM3 have very mixed results, and if you look at the literature, TIGIT and TIM3 are actually not very good checkpoint inhibitors to target when going into the drug, the drug manufacturer for actually curing cancer. So PD-1 is a very effective one, as is ICOS. And this dovetailed quite nicely with the established data. Looking at CD8 positive cells, very similar results, that PD-1 was significantly increased, as was ICOS. And again, TIGIT and TIM3 showed much less expression, in TIM3 actually being downregulated in the tumor microenvironment. So this went very straight to the heart of the movies I showed you before, that when you expose the T cells to tumor microenvironment, there are massive changes in the metabolic, cytokine, and checkpoint receptor profiles of these cells. And it is imperative to take this into account when you're making a CAR T or any cell therapy, that the cells need to be prepared to go into that tumor microenvironment. And as we all know in cancer biology, the tumor microenvironment is very immunosuppressive, which is why tumors can grow so well. So it was very exciting to them, and they are continuing their work. And actually, I believe this is going to be published in early 2019 with additional data that they did not share with us. So I will go on from there to the next case study, again in immune cells. And this was actually performed in Xcell Biosciences, looking at NK-mediated tumor killing efficacy. And as you see here, we had the NK cells, which were from normal healthy donors that were expanded for 7 days under the 2 psi and 2 psi and 15% oxygen versus no pressure and the same oxygen, more than 15% oxygen. And after the culture for 7 days, the cells were co-cultured with DU-145 prostate cancer cells. And then this was performed for 24 hours and then looked at the cell killing on a bright field. As you see here, this is our standard incubator, ambient O2. And you see here that 36% of your tumor cells are remaining. So the bars actually represent the tumor cells, not the NK cells. And so the standard incubator yielded 36% of the tumor cells remaining. However, when you raised the NK cells in the atrial vasculature tumor atrial vasculature microenvironment, you had a significantly increased killing of the cancer cells. And again, showing that the NK cells, if you put them into their essentially native microenvironment, the killing and their performance as NK cells actually improves. So as I said, one of the other areas that we have done a lot of work in at Xcell Bio is with stem cells. And so this was actually work done by Zach Papalardo at Xcell, and it's fibroblast reprogramming and a neural differentiation workflow. And as you can see here, it is a complete study that goes from skin fibroblasts all the way through to iPSCs, to neural epithelial cells, all the way down to mature either motor neurons or CNS cells. And I'll show you that in just a minute. So the first part we are going to do is look at the reprogramming. And the way Zach did this is we did 5% O2 plus or minus pressure and compared that to the oxygen and pressure only atmospheric in a normal incubator, and then look to see the iPSCs that were produced. They wanted to take a look at how this would work in different cell lines from different sources. And as you can see here, the gender is mixed, and also the age of the donor is also highly variable. So they were just testing over different sources of material. Does this 5% O2 and pressure work? And as you can see in the green bars, basically for most of the donors, all but one, in fact, the hypoxia 5% oxygen and pressure actually gives you more colonies per 40,000 cells transfected. So it was from this that they moved forward and did 3 independent reprogrammings, and you see from this data that, again, the oxygen and pressure at day 16, 19, and 22 gave you a significantly increased fold change in the colony number. And as you see here on the right, 3 telltale, actually 4 telltale markers, NANOG, OCT4, and SOX2, actually I think it's just SOX2, are increased. So again, indicating that these cells are truly in a pluripotent state. So using the avatar for induction patterning and improved efficiency, this was again performed at Xcell Bio in our headquarters. They used a neural induction medium at 5% oxygen and 2 psi versus neural induction media in a regular incubator. As you see, the standard incubator here, you have very low PAX6 DAPI nuclei ratio, but when you add the hypoxia, you get a bump, and then when you add the hypoxia and pressure, you get a further increase in the amount of PAX6 being expressed, which is an indicator of neural induction. So you see here whether the cells came from adult iPSCs, iPSCs from adults or iPSCs from neonatal source. This works well in both instances. Again, indicating that pressure was one of the vital components to actually get increased expression. To bring this all the way down to terminal differentiation and fruition, we see here that so the CB is cord blood and BM is bone marrow. So these are neural stem cells derived from cord blood iPSCs and neural stem cells derived from bone marrow iPSCs. And you see with the 2 psi and the hypoxia, you get much better staining for these different markers of mature motor neurons or mature CNS type neurons. And you see here, this is the quantitation of that. Again, the 5% O2 and 2 psi actually showing a definite increase in these key markers. So it shows the utility of the avatar with the hypoxia and pressure at different settings for each one of these steps to go all the way through to get you from the reprogramming all the way through to the mature either motor neurons or CNS type neuron. We also, as I indicated at the beginning of the talk, the company was founded to accommodate tumor cell growth and expansion coming out of primary tumors within the from human patient samples, and having an avenue in the culture system to actually express them and have them express their clusters of differentiation. So one of our customers, Immunocore in the UK, a very nice statement by our collaborator there. I've never seen a product work as well as promised by the manufacturer. So their problem was the inability to maintain and expand primary patient tumor samples for drug screening. They wanted to fine-tune the oxygen and pressure in our system to mimic the tumor microenvironment and expand non-small cell non-small cell lung cancer and breast cancer from patient-derived tumors. And at the end of this, it enabled the expansion of tumor cells that could not be grown before. And the Avatar is currently integrated into their drug screening workflow for the target primary cells, a source for those. And the core facility is now expanding those capabilities to blood cell expansion. So again, a very nice story for Avatar, that using the appropriate settings for the appropriate microenvironments allows you to grow cells, more of them, and they have better indicators of their maturity and so forth. The final study I want to take you through is looking at Palacký University in Olomouc, the Czech Republic, and this is work done by Dr. Das's group at Palacký University. And they were interested in determining the effect of hypoxia and pressure on cancer cell lines. Specifically, they wanted to monitor the markers for epithelial and mesenchymal transition. This is a model for migratory and invasive capacity. Essentially, they're looking at metastases. So the epithelial cells are attached and polarized, and as they transition towards the mesenchymal cells, which are unattached and migratory. Therefore, they're able to basically metastasize. They were using HCT116 and HeLa cells. These are a cancer, colon cancer cell line, and these are a cervical cell line, as you probably know. And they were prepared in 4 different conditions. So the standard incubator is atmospheric oxygen and 0 psi, atmospheric oxygen and atmospheric pressure. Hypoxia chamber was at 1% O2 and no pressure. And then the Avatar condition 1 was 5% O2 and 2 psi. And 2 psi actually corresponds to normal blood pressure. So this was actually a logical choice for the 2 psi above atmospheric. And then the Avatar 2 setting was with the hypoxia and the 2 psi. So you'll see direct relationship between pressure hypoxia with or without pressure, and showing the effect of pressure. And the cells were handled in these conditions here. As you can see, cell viability by day 3, you saw that the colon cancer cells, the HCT116s, were affected with the hypoxia and pressure, basically a little bit of decrease here. But interestingly enough, the opposite basically happened with the HeLa cells. The cervical cancer cells actually did better with hypoxia and pressure versus the normoxia. And it did, showing the utility of pressure. The hypoxia chamber is at 1% O2, as is Avatar 2 setting. but the only difference between these two is actually the 2 psi. So again, pressure definitely matters in this situation. The first thing they wanted to look at was vimentin. Now, vimentin is increased in the mesenchymal transition. It is allowing the cells to move towards that mesenchymal, and then thus the metastases state. And what you see here is the HeLa cells derived either from 2D or spheroid-derived culture. When they are put into the Avatar 1 condition, which is 5% O2 and 2 psi, you see actually a fairly high increase in the vimentin expression. And when they're put into the avatars, the second avatar condition, which is 1% O2 and 2 psi, again, you see a significant increase in the vimentin expression when compared to the normoxia or hypoxia alone. So again, it comes to the conclusion that the 2 psi actually matters in this case for vimentin expression. E-cadherin was also looked at. E-cadherin is an adhesion molecule which is downregulated in the conversion from epithelial to mesenchymal. So we should expect very little to no E-cadherin expression as these cells go towards that. And in fact, that is what you see. Only lane 5 had E-cadherin expressed, and that's the colon cancer at 1% O2 and 2 psi. But by and large, especially the HeLa cells, had absolutely no expression of E-cadherin. Beta-catenin. Beta-catenin is also required for the epithelial to mesenchymal transition phenotype, and it is shown very increased in the HeLa cells, and as compared to the normoxia or hypoxia alone. CACO2 cells are a colorectal adenocarcinoma cell line, which are actually a model for the epithelial cells. And these are your positive control, again, showing the beta-catenin expression. So again, the conclusion from this was that the avatar was having an effect compared to the normoxia and the hypoxia chamber alone in increasing this epithelial to mesenchymal transition state. They then went on to look at CD44 and CD133. These are markers that are used to track the resistance to chemotherapy for colorectal cancer. And what they found was there was essentially no indication that CD44 is increased in the HeLa cells here. The CACO cells definitely have some amount of expression here. But again, for the CD44 and the resistance to chemotherapy, there was no increase in that using the avatar conditions. They then went on to look at the influence of pressure on lentiviral transfection. And this is looking at normal pressure, so no PSI, and this is above 15% in a standard incubator. And then the avatar, which has 2 psi. And what we saw was that there was an increase in the amount of fluorescence intensity seen here as opposed to the normal pressure under the standard incubator. And what this worked out to be is basically a 28% increase in with the pressure in the transfection using lentivirus. So again, showing that pressure does help your cells in different things that you're working with. It will help you drive either transfection or expression, and they found this actually quite useful. So the results are, in summary, are seen here. Basically, the avatar control system resulted in an increase in the epithelial mesenchymal transition phenotype. It did not help the expression of the chemotherapy resistance stem cell markers. However, it was quite useful in the efficiency of transfection. So with that, I will entertain any questions you have.
Wonderful. Thank you so much, Greg. That was very informative. Some really great examples. We do have some questions that have come in. The first one, what is the proposed mechanism for improving viability of cryopreserved cells?
That is actually one of the case studies I actually did not handle today. That is actually a very interesting case study that we have. It came from the NCI, and basically they do 5% O2 and 2 psi. And the thought processes, and we don't quite know what that is today. We haven't investigated this closely, but the idea is that you put them under 2 psi, and that actually stabilizes the cell as they're coming out of the freeze thaw process. So that's the idea of why pressure is working, why pressure is important for the cryopreservation.
Excellent, thank you. So in this picture here, I see 4 units. Do you sell them as 4? Why not just 1 unit?
In general, we do not. We actually do sell them in units of 2, so that's the duo. What you're seeing is the AD system, and that is a 4 system. In general, we don't sell them as single units, and the reason for that is, as most of you kind of learned in this talk, a lot of this is very new, and there's much more that we don't know than we do know. And most customers want to have multiple units so they can optimize those settings, in parallel as opposed to sequentially. So if you have one, you test out one setting and then have to change and then have to go to another setting. This way, with at least 2, you have 2 units that will allow you to test side by side, and you have your control basically right there with 2 settings, basically on the same cell line or cell prep. So that's essentially why we don't try not to sell them as single units.
Okay, thank you. How long has the Avatar system been on the market?
The Avatar was first commercialized January of 2017.
Okay, and do you have systems placed throughout the world?
Yes, we do. So we have systems from the UK. We're actually some of the first in the UK, the United States, of course, China, Saudi Arabia, and into mainland Europe.
Great. Another question I see here, how is pressure modulating cellular metabolism?
That's a very interesting question. So we know a little bit from RNA-seq data that the pressure actually affects the cells through the cell membrane and the cytoskeleton. How that exactly happens, we are not entirely sure, but the one thing we do know, depending on your settings with the hypoxia and the pressure, it can affect the cells in different ways, and one setting may upregulate certain genes and downregulate others, but this is not always the same for the same settings. So the so basically it depends on the context that you're in, is the oxygen and the pressure. So whether those certain genes are upregulated and then downregulated.
Okay, thank you. Another question. What impact does pressure have on dissolved oxygen levels in the medium?
We have a lot of theories about that, and we are actually about to embark on a testing program for that in San Francisco to work on that and to measure that directly. We have a lot of theory from physics papers, but we want to measure that directly, because once you introduce either media, salts, serum, it gets very complex, and at this point, right now, we need to do a direct measurement.
Wonderful, thank you. So that seems to be all the questions we have. Thank you all. We will be putting a recording of this up on our website at xcellbio.com. Thank you to Greg, and thanks to everyone for attending.
Thank you very much.
Thank you.